Topo cloning technology revolutionized cloning to deliver unparalleled simplicity, efficiency, and speed. No matter how difficult the sample or downstream application, 95% recovery of your desired clone is possible in just 5 minutes. But just how does this technology work? Let's start in the lab, where we'll find our research scientist Jeff setting up a topo cloning reaction. He combines his PCR amplified product with the water, salt solution, and topo vector, all included in the kit, and he then mixes gently. While Jeff incubates the reaction for 5 minutes at room temperature, let's take a closer look at what's happening in the reaction. The key to topo cloning technology is the enzyme topoisomerase 1, which functions both as a restriction enzyme and as a ligase. Its biological role is to cleave and rejoin DNA during replication. It binds to the phosphate group attached to the 3' thymidine and cleaves the DNA strand, enabling the DNA to unwind. The enzyme then religates the ends of the cleaved strand and releases itself from the DNA. To harness the religating activity of topoisomerase 1, our topo vectors are supplied linearized and have topoisomerase 1 covalently attached to the 3' phosphate group at each end. When the topoisomerase 1 activated vector comes into contact with Jeff's linear DNA fragment, the DNA fragment is ligated into the vector with high efficiency, and the topoisomerase 1 enzyme is released. In this experiment, Jeff has amplified his DNA of interest using a proofreading polymerase with 3' to 5' exonuclease activity, because the accuracy of his sequence is crucial to his application. These polymerases generate blunt-end PCR products, so he is using our zero-blunt Topo PCR cloning kit. It is not necessary to purify the PCR product prior to topo cloning. As with any PCR cloning technology, the cleaner your PCR amplified DNA, the higher the likelihood that you'll clone the correct DNA insert. Some researchers use a Taq polymerase that generates PCR products with 3' A overhangs. In this case, a Topo TA cloning kit must be used, as it includes a vector with complementary T overhangs, allowing the insert to ligate into the vector effectively. After incubating for 5 minutes at room temperature, the ligation is complete. Jeff is now ready to transform E. coli with the topo vector containing his DNA fragment of interest. He adds his topo reaction to a tube of competent cells and completes a few short steps to allow the cells to take up the vector. He then plates the cells and allows transformants to grow. Since the cloning reaction is 95% efficient, Jeff can have high confidence that the colony he picks will contain cloned DNA. He also has the added flexibility of using competent cells that either come with the topo cloning kit or are sold separately. As you can see, Topo PCR cloning technology is a fast, efficient, and easy way to get the cloning results you need for your downstream experiments. Backed by over 20,000 citations, Topo is a brand name you can trust for even your most difficult to clone samples.